Hello, I'm Terry Jernigan, and I'm the co-director of the ABCD Coordinating Center. And my job today is to just give you a brief introduction to the ABCD study, which is the largest long-term study of brain development and child health in the United States. The ABCD Research Consortium consists of a coordinating center, a data analysis and informatics resource center, and 21 research sites across the country. And I'm going to start by talking a little bit about the original rationale for ABCD. As everyone in this audience knows, both the extramural community and the leadership at NIH has appreciated for a while that neuroimaging research reveals a very protracted period of ongoing biological development of the human brain throughout childhood, all the way through adolescence and into early adulthood. And it's also been appreciated that adolescence is really a pivotal time in the lives of many young people. Under the best circumstances, it's a time of rapid increase in their knowledge, their skill, and creativity. They become more engaged with their peers and with their futures. They become more independent and usually more resilient. But it's also, unfortunately, the time when the earliest signs and symptoms of some serious mental disorders first appear. And it is, without any doubt, a time of increased risk-taking. And this can be adaptive. It gets young people out of the comfort and safety of their nuclear families to engage and sometimes compete with their peers and to find their mates and to explore. But of course, this heightened risk-taking also brings increased risk of serious accidents and dangerous experimentation with addictive substances and behaviors. So it's been known for some time that variability in these important outcomes in health, mental health, social and academic spheres can be associated with genetic, environmental, and behavioral factors. But clearly, more effective interventions to prevent and protect against adverse outcomes will rely upon better models of the specific roles of these different factors. And unfortunately, again, it was understood by the extramural community as well as the leadership at NIH that because of limited scope and size and sometimes the method sensitivity of previous studies, there's been a reduced impact of the research done before. And so the idea for a large-scale study of the adolescent brain originated actually at NIDA and was developed in this collaborative entity called CRAN, the NIH Collaborative Research on Addiction entity, which is a collaboration between NIDA, NIAAA, and the National Cancer Institute. And then as the planning proceeded, a number of other institutes and centers recognized that ABCD provided an opportunity for them to address some of the questions that they were most interested in. And so at this point, ABCD is supported by 10 institutes and centers of the National Institutes of Health and 4 additional federal partners, the National Science Foundation, the CDC, the National Institute of Justice, and the National Endowment for the Arts. Now, this is the final consortium configuration for ABCD. You can see there are 21 research sites, many different elite institutions all around the country. You can see there's a coordinating center and a data analysis and informatics and resource center. But I want to draw attention to this gray box because this study is conducted as a set of cooperative agreements. And that means that we're working very closely and in collaboration with our federal collaborators. And we have a robust complement of active research partners from the institutes and other agencies. Some are assigned full-time and many part-time to ABCD. And they work right alongside us, right alongside all the rest of us in the consortium. I want to particularly mention Dr. Gaia Dowling, who's the NIH project director of ABCD, but they bring their expertise and the interests of their home agencies to our ongoing decision-making processes. Altogether, there are about 150 active full investigators working on ABCD, and this includes collaborating federal researchers, and we all play roles in decision-making. In addition, there are 35 postdoctoral associate investigators working with us. This just shows you sort of the geographical scope of ABCD, where we're located. And as you can imagine, our study aims have broadened a bit as our partners increase, but we do expect to have an opportunity to develop national standards for the first time for normal brain development. We'll be measuring individual developmental trajectories of brain, cognitive, and emotional development, academic progress, and trying to identify factors that influence these trajectories for good or for ill. We'll be trying to examine the roles of genetic versus environmental factors and the constant interplay that we expect to see throughout development. We'll be studying effects of health, physical activity, sleep on brain development and other outcomes. Because of the size of our cohort, we will be observing the onset and following the progression of mental disorders. We'll be looking for variability in the course or severity of these disorders, and we'll be examining the relationship between mental disorders and substance use. And then back to where we originally started with the study, the ideas for the study started, we'll certainly be looking, a very important priority for us is to try to determine how substance use exposure affects developmental outcomes and vice versa. So here's a quick overview of the study as we are today. We did enroll 11,880 young people aged 9 and 10 throughout the country, and we will follow them for at least 10 years. We used a school-focused recruitment strategy, which was guided by an epidemiological team, and we included in that a twin cohort that was recruited from twin registries. And you're going to hear a lot more about the assessments and the different modalities and domains of assessment in the presentations that follow. But just to give you an overview, we obviously do multimodal neuroimaging. We do extensive health and behavioral assessments. We try to track the activities and monitor the environments as much as we can of the youth. We try to collect as many informative biosamples as we can. We're interested in hormones, puberty, genetics and epigenetics, and substance use exposure. And you're going to hear about some novel wireless assessment technologies that we're trying to employ to improve our phenotyping of the sample. Okay, we've recruited a very diverse cohort. Largely, we have been able to match the targets that were set for us for a cohort composition on race and ethnicity that were set for us by our epidemiological consultants. And this just shows you that in what we refer to as our general population enrollments, you can see here in this dial that in the outer ring of this dial is the number of individuals, represents the percentage of individuals we recruited from each of these ethnic groups, racial groups. And the inner dial shows you the targets that were set for us by the epidemiological team led by Steve Jernigan. And this is just showing that we did almost as well in our twin registry. So this is the twin cohort. These were recruited differently through birth and twin registries at only 4 sites. But they also did quite a good job, not quite perfect, but a pretty darn good job of recruiting a very diverse set of twins. Now, one thing you need to know about ABCD is that there's a lot of relatedness in ABCD. And in fact, we're still trying to figure out exactly how much relatedness we have in ABCD. But this is kind of a rough summary of it. Over 8,000, 8,150 kids, probably at least, are mostly single birth kids recruited from the general population. 730 kids were recruited from twin or birth registries with their co-twins in same-sex pairs for the twin cohort. Over 1,700 children are either half or full siblings, but are not same-sex co-twins in the sample. Now, quickly, the visit schedule, this is just a basic overview of the visit schedule. At baseline and biennially, every 2 years after that, we do very comprehensive assessments, which include the multimodal neuroimaging that I'm sure you'll be interested to hear about today. We do, normally we do face-to-face visits at the interim years, that is the odd years in between those biennial visits, but briefer without the imaging and and also pared down in some of the other modalities as well. At the moment, we're not doing any face-to-face visits. All of our visits are virtual, of course, because of the epidemic. And then we do more frequent phone and web assessments at least every 6 months in the normal visit schedule. Now, this is the I referred earlier to a kind of distributed decision-making process in ABCD, and this graphic shows the consortium's organizational framework. And I think it reflects that and gives you some idea how we do things. Obviously, as we attempt to ask the right questions, select the right assessments, take advantage of emerging scientific opportunities, we also have to monitor the performance of our instruments and address gaps and weaknesses. And very importantly, our youth are growing up, and they are growing up in a rapidly changing cultural and technical environment. And this means that we have to constantly be adapting our protocol to ensure that it remains developmentally well suited to our cohort and that it makes meaningful contact with the salient aspects of a changing environment. For example, we recently had to adapt our protocol and our study quite dramatically when we had to pivot to addressing the impact of the COVID-19 epidemic. So this is an example where we had to virtualize our assessments, and we had to figure out how do we get the most critical information to assess the impact of the epidemic itself on our cohorts and how it's maybe changing the trajectories that we're measuring. And we do this with work groups, which are, and you can see many of them listed here in every domain of assessment. These are experts who help to develop proposals, followed by integration and implementation in the centers. And before I end, I want to draw attention to the fact that ABCD is conducted as a very much a leaning into an open science model. And it has a pretty aggressive sharing timeline. As many of you know, we share the imaging data, the raw imaging data on the fast track within about a month. But most of the data is curated and released annually. And we had the 1.0 release was in 2018 with about half of the baseline data shared at that point across all domains. And the 2.0 release was in 2019. And this pretty much wrapped up the baseline data set. And importantly, in the next one, which is the 3.0 release, which is coming out sometime between now and the fall, I think, we will be, for the first time, sharing longitudinal data in the comprehensive protocol. So our year 2 data, which is the first follow-up, full comprehensive follow-up, will include neuroimaging data, for example. So this will be the first time we'll have longitudinal neuroimaging data. We expect that to be present in 3.0 in over a half of the cohort. So let me thank you for your attention, and I'd like to acknowledge the many partnering federal agencies that I listed earlier, and the magnificent ABCD consortium, many of whom are pictured here at one of our most recent annual meetings. I also want to remind you to send your questions to this site so that we can address them in the question and answer period. Thank you again for your attention.
And my name is Susan Tapert. I'm from the University of California, San Diego, and I'm here to talk to you today about the physical, behavioral, and environmental phenotypes in the data in the ABCD study. ABCD data is vast, and it's organized into data ontologies that help users of the data to navigate the large amount of behavioral phenotypes that we have available in this study. We have data on youth's neurocognition, substance use, culture and environment, physical health, gender identity and sexual health, genetics, geocoding, as well as mental health and novel technologies that you'll hear about in other presentations in this set. The study design of ABCD is that there's an initial baseline assessment, annual follow-ups, and then there's a brief mid-year assessment. At the baseline age, participants were 9 to 10 years old. 1-year follow-up, they're 10 to 11 years of age. The 2-year follow-up, they're 11 to 12 years of age. And these are the time points of data that are available in the 3rd data release of ABCD. This is an example of the data ontologies for the neurocognitive data in ABCD. And if you look to the far left of this tree, you see ABCD, and then it's broken down into the main study of ABCD. There's another lower branch for data that pertain to smaller sub-studies that occur within the ABCD study. So in the main study, we have kind of those sections of data that I have listed, which include neurocognition as an example. And neurocognition is then broken down to the different neurocognitive tasks that are administered in ABCD. And each of those tasks has a large number of variables available for your use, reflecting youth's performance, things like accuracy and reaction time. And some examples of these variables are shown on the right side of the screen here. I want to review with you the specific measures that are used in ABCD to assess neurocognition of youth, because we want to study this over time. So we have measures that we collected at the first time point, the baseline time point, a few different measures that we alternate in on the odd numbered years, the 1 year follow-up, and then also at the 2 year follow-up, we have another set of tasks we use that we can use in direct comparison with the neuroimaging data that are collected on the baseline, 2 year, and then moving forward on the even numbered years. So we collect a vision screener with all of the kids. We assess their right-left handedness. We use the NIH Toolbox cognitive module and have 7 tasks in ABCD that we've used from that module. We assessed learning and memory with the Ray Auditory Verbal Learning Test through Q Interactive, another Q Interactive task with the Matrix Reasoning task, and then we use a group called Millisecond to help develop computerized tasks that measure kids' performance on a range of other skill sets. So we have a visual spatial task known as the Little Man task, cache choice and delayed discounting, which look at their kind of ability to delay gratification, emotional Stroop task, which is getting at their ability to kind of inhibit prepotent response, and it has other affective components. Game of dice, an executive functioning decision-making task, and a social influence risk-taking task to look at the degree to which their decisions are pulled by how peers make choices. Moving on to the next domain in ABCD of data is substance use. We're very interested in ABCD on young people's use of substances. And at this point in the study, we're really focused on how kids initiate substance use. So looking at our ontology tree again, we have ABCD and its main study, the primary domains that I've mentioned, and then substance use has this kind of a deep array of measures, including things focused on their alcohol use, caffeine use, and use of other substances. And we ask some additional questions and measures for those kids who have endorsed some substance use to understand more about those particular experiences. For all of the kids in the ABCD study, these are the kinds of data that we collect concerning substance use. We have some biospecimens that look at recent use of substances and past 3 months of substances that come from a hair sample that's analyzed in a lab for a portion of participants. And then we ask everybody about the recency of any use, kind of a detailed interview that goes into any substance use experiences. And then we ask some broader questions about how their peer group tolerates reports of substance use, peer group deviance, including peers' involvement in substances, their intention to use for those who haven't used yet, how risky they think use of various substances are, and what kind of effects they think would be produced by using substances those are known as expectancies. These questions are asked annually of the youth. And then for those who report having some use experiences, we provide some additional questions. So if they report having sipped alcohol, we ask some additional questions about that. And if they reported having 2 or more uses, there's some additional questions that we ask shown here. And we would also ask the diagnostic from the diagnostic interview, the KSADs, we ask the alcohol use disorder and the drug use disorder modules. We ask the parents some questions about the youth substance use as well. So for every child who is participating in the ABCD study, they also have a parent or guardian who we ask to participate as well. And they are both interviewed privately and separately to ascertain these data points. The parents, we ask them to the best of their knowledge about recent use of their child in the study about substances. We ask about parent rules on substance use in the household, community and protective factors, and then also some questions about substance use in the household and how substances of abuse, including prescription medications, how those are stored in their household. We do some mid-year assessments in the ABCD study. So, we talk to participants in person, usually in the lab, but at this point during the coronavirus pandemic, we're assessing them virtually through Zoom and telephone. But at all times in ABCD, we assess the youth at the midway point between these annual assessments. And we let the youth choose if they would like to be interviewed by the phone or if they would like to self-complete the forms that we send through a link in REDCap. We assess youth substance use here in these interviews, and there's other mental health symptoms that are assessed as well. Culture and environment is a very important aspect of the ABCD study. And in the culture and environment kind of ontology, you can see a listing of the primary measures of culture and environment that are part of the ABCD protocol over our various time points or waves of assessment. And on the right, it is just a kind of an example of some of the specific variables that are available and how they're described in the data dictionary, and we have additional reference information that is provided as well, so you can look up and see where the measure comes from and look at the psychometrics of the measure. This is a list of the questionnaires that we give in ABCD to youth in this domain of culture and environment at the baseline, 1-year follow-up, and the 2-year follow-up. We're asking things about acculturation. We're asking a lot of questions about family dynamics here as well as another important part of youth environment, their perception of the safety level of their neighborhood. We ask about parent monitoring. We ask peer group behaviors too. Also, we ask youth about their perception of feeling discriminated against based on race, ethnicity, religion, body type, and other measures as well. We ask about the school factors, including kids' attendance and their grades, and perceived risk and protective factors of their school, as well as their ability to cope with different problems that might come up. Parents are asked sometimes overlapping and in some cases different questions about the youth's culture and environment. Family cohesion, family environment are very important. Also acculturation and cultural values. We use the Mexican American Cultural Values Scale, but this is given to all of our participants, not only our Mexican American participants. We ask about kind of ethnic identity as well, and another measure of acculturation. And then a little bit about safety and crime perceived in the neighborhood and school. Here are our physical health measures in ABCD. So physical development is very important in this age of adolescence. We have self-report from the youth, parent report from the data on different facets of physical health, and we also have some biospecimens that fall under this umbrella as well. So you can see here in our ontology under physical health, an array of measures that we collect. And an example, as we kind of magnify in on an example here with one of our sleep measures, the kind of variables that we have available. This is a list of the measures in the physical health domain that we give to youth in the study. So we measure kids' height, weight, their waist circumference using CDC approaches for these measurements. At the first time point in ABCD, we collected a saliva sample for DNA analyses, so we can do genotyping and other kinds of genetic analyses too in the future. Starting at the 2-year follow-up, as the kids are a little bit older, we have started to do blood draws with the youth so that we have a little bit more flexibility in the kinds of genetic analyses that will be available And we're also able to look at some other health factors that can be gleaned from a blood sample. Of course, if a child does not want to have a needle in their arm and doesn't want to give a blood sample, not a problem. We just carry on with the assessment. But more than half the kids are willing to do that. Blood pressure we collect as well on the kids. We also collect annually a saliva sample so that we can look at pubertal hormones. We collect self-report data on pubertal development to help stage with Tanner staging where the kids are in their pubertal development. For girls, we ask questions about where they are in if they've had their first period, where they are in their menstrual cycle. Sleep, nutrition, pain, and physical activity are other key domains that we're collecting as well. And we begin to ask the questions of the youth a little bit more as they get older. We ask the parents quite a few questions about the kids' physical health, as shown here, including a pretty detailed interview of the youth's developmental history. This includes prenatal exposure to different substances, when kids achieve various developmental milestones, and then a medical history questionnaire where we're asking questions about the kinds of medical issues the child may have experienced. Separate form where we're collecting detailed information about medications that the child has used. We collect from the parents any information they might have on the youth's menstrual cycle, if they were a girl at birth. And then we collect information from the parents on the youth's nutrition, the kinds of foods they eat, any kinds of history of traumatic brain injury, an additional confirmation about pubertal development, sleep, the kinds of sports activities kids are involved with. And then we also ask parents if they have any baby teeth of the child at home that they'd be willing to bring in. These are being saved up for subsequent analyses to look at things such as exposure to various environmental toxins. ABCD has a gender identity and sexual health work group to help make sure that really the wording throughout ABCD is careful and appropriate with regards to gender. And here we also have some additional measures that ask about youth's gender identity, and then some other questions that ask about sexual experiences in an age-appropriate manner. So here are some of the questionnaires, some of the data that kind of fit in with these items. And the specific measures are listed here for the youth and for the parents. Okay, so we also have for some genetics information that is available in ABCD, quite a bit of genetics information. This is kind of under one of our biospecimens that we collect because the data originate from a saliva sample or a blood draw. And at this time, we have quite a bit of genetics information available in the upcoming data release, available in several different kinds of formats. We have been able to collect genotyping data on the vast majority of ABCD participants. We have multi-ethnic reference panels for imputation with TopMed reference panel. We also have CNV calls, and then we have in the tabulated data release, genetic ancestry factors. Also, ABCD has a large proportion of twins within our cohort. We specifically designed our study in this manner to have a large number of twins. And so in our genetics data, we have zygosity for the twin samples. To try to reduce the burden on the participants of asking them too many questions, we also make use of geocoding approaches to learn more about our participants. What we do here is we collect residential history information from our participants. We ask this of the parent, and we ask this of the youth's entire history of in their life, where all they have lived. We do not share any address information with anybody in data releases. This is completely confidential information, but we are able to take this residential history information and take a look at other available databases and gather specific information about the youth's environment And that is what you'll see in the database. So based on the current and the past residences of the child, we cross that information with publicly available geospatial data. And we're able to get things from census-defined urban categories. And so for one example is we are able to get air pollutants, traffic counts, and just to emphasize that no address information is saved, but we have this other information based on the use locality that is included in the data. I just want to make sure that you're aware that we have this wonderful resource in ABCD called the DEEP, the Data Exploration and Analysis Portal. You can see the link to access this here. And the DEEP is a way that we can provide advanced statistical analysis functions so that you can work with the most recent ABCD data releases in a really effective manner. DEEP can suggest some optimal covariates and models for you to use based on the kinds of analyses that you have in mind. You can save your syntax and output, and this can also save you from needing to download all of the data onto your own hard drive, but rather you can do a lot of the analyses in the cloud, save your syntax and output, and then modify your syntax and employ that to run, slightly tinkered with analyses to help you get the results that you're looking for. Access is available to users who have current access to the ABCD data, which, as you may know, is available through the NIMH Data Archive. The link is here. And through that process that you would follow through this link, you would request an NIMH Data Archive data use certification. Takes a couple of weeks to get that, and you will go through your institution, your university or the research institution where you're based, and then you'll use your NDA credentials to log in and begin exploring the data. So please send your questions to this link. And we're just so grateful for our ABCD families, the kids and the parents who take the time to participate in the study. And we're very grateful to our funding support through the National Institutes of Health and other federal partners. Thank you so much for your time and interest today.
Hello, everybody. Thank you for joining me for this part of this educational course. I'm going to be speaking about the mental health assessments that we are collecting as part of the ABCD study. And before I start, I really want to take a minute to acknowledge the entire ABCD mental health work group who have been a huge part of designing this component of the study. And in particular, I want to shout out to Alexi Potter, who is the current co-chair of the mental health work group, and Ken Scher, who was the previous co-chair, and Susan Tapert, who has been our liaison with the coordinating center. And I'll just start by disclosure of interests, of which I don't have any conflicts. And I also want to start with a thank you and a huge acknowledgement for all of the ABCD families and child participants. None of what I'm going to be talking about today and none of the data that's available to you in the community would be possible without our wonderful participants. So let me start by talking about the principles that we used when designing the mental health workgroup assessments for the ABCD study. First, we really wanted to use, where possible, instruments that already had documented reliability and validity. In some cases, we were not in a position to be able to do that, but where possible, we definitely selected things that already had established reliability and validity. Second, we focused on using developmentally sensitive and appropriate instruments, thinking about both where our participants were at each phase of data collection and what sorts of things would sort of stand the test of developmental time. We also wanted to ensure that we engage the most appropriate informant depending on the developmental stage and domain. And I'll say a little bit more about that as we go along. We also really wanted to try to minimize participant burden. There are so many different things that we wanted to find out, but we only have so much time, and we really want to make sure that this is a positive experience for our participants, so we really focused on minimizing participant burden. And we also wanted to be informed by the prior relevant literature, as as to what the best measures were and what things were going to be most informative going forward. And we are always, this is sort of an ongoing balance and sort of tension between consistency over time, meaning measuring the same thing with the same measure over time, and capturing new behaviors and outcomes as they become developmentally appropriate. With these kinds of studies, you can't just keep adding and adding and adding because the participant burden becomes overwhelming. But there are things that you might want to capture when the child is a teen that you really weren't able to capture when they were younger. So as we have gone through the different waves and are planning the future waves, we're really always thinking about this balance between consistency and capturing new behaviors or outcomes, or things that become more developmentally appropriate as the child moves into a different developmental stage. So let me first just give you a broad overview of the timeline. I know you will have seen this before by now, but I think it's useful for thinking about what assessments we're doing when. So these are the sort of different major categories of assessments. And then here are the different waves: baseline, year 1, year 2, year 3, and year 4, which are all in-person waves, modulo COVID. And then 6 months, 18 months, 30 months, 42 months, which are phone or online assessments. And let me pull out in particular the mental health assessments to give you kind of a finer grained overview of those assessments. So we really have parent report on youth, youth report on themselves, parent report on themselves, parent report on the family, and we also actually have teacher reports. And so these little computers give you a sense of when these different kinds of reports are collected. So at baseline, we have parent reporting on the youth and themselves and the family. They actually will continue some of this self-report in the out years. We then have youth self-report at pretty much every wave, whether that's an in-person wave or a phone online wave, although the nature of that report differs a little bit depending on the wave, with the phone, for example, or online assessments being much shorter than the other assessments. So this just gives you a broad overview of the timing. Let me talk a little bit about the different domains that we're assessing. So we are assessing youth and family mental health diagnoses. So think of this as categorical assessments of whether or not a child or a family member meets criteria for a particular diagnosis, but then also much emphasis on dimensional mental health measures. So getting assessments of dimensions of symptoms or dimensions of behaviors that are mental health relevant. We are also trying to capture personality and other quote unquote trait-like behaviors that are known to be relevant to mental health or substance use. And then we're also trying to capture other things that are very relevant for thinking about mental health, like stress, life events, friend relationships. Let's go back a minute, because we know that those things are going to kind of critically interact with mental health over time. So let me start by talking about mental health diagnoses. So when we were designing the ABCD originally, we spent a lot of time trying to figure out how are we going to be able to capture diagnostic information about the youth in particular. And the tension there was that it was really not going to be feasible from a practical or a financial perspective to do clinician-based diagnoses of every child at every wave. We also really wanted to be able to capture a current diagnostic criteria, meaning DSM-5, as opposed to DSM-4, DSM-3 criteria. And so when you put those two considerations together, there were not, and we also wanted something computerized, because when you're assessing, 11,800 plus kids, we were not going to be able to do something sort of, on paper and then transcribe it. So when you put all those considerations together, we didn't have a lot of options. But fortunately for us, Joan Kaufman and colleagues, Ken Kobach in particular, had just developed a version of the KSADS that they had computerized for DSM-5 diagnoses. And they had created what we'll call self-report versions for both parents and youth. So that is what we settled on in order to be able to get diagnoses. So we have the parents report on youth using the computerized KSADs, and that started at baseline and has continued forward. So that is, to be very clear, not a clinician administered instrument. It is the parent sitting down at a computerized version of the KSADs and responding about what they've seen about the child. It's computer administered. Like I said, it's designed to capture DSM-5 diagnoses. And we focused on mood disorders, anxiety disorders, psychotic disorders, the sort of range of externalizing disorders, including ADHD, ODD, conduct disorder, and suicide. And I'll show you kind of the design over time, so you can see what parents are reporting on at different time points. So far, only parents have been reporting on the externalizing disorders because of the data suggesting that self-reports on this are not particularly good really any time in the life, but definitely not in kids, and that external reporters are going to give you more accurate assessments of these kinds of externalizing diagnoses. We then also have the child self-reporting on themselves on the computerized KSADs. Now, the computerized KSADs self-report was designed for 12 and older. And so when we started the ABCD study, we did not think that the children were old enough to really do this on their own. So in collaboration with Joan Kaufman, we implemented this as an R research assistant assisted child self-report on the computerized KSAD. So the research assistant will sit with the child and help them go through this. And we think that has been important. We really don't think that the children could have done this on their own at this age. They are now getting to the age where it's much more feasible for them to do it on their own. But at baseline and year 2 and year 1, we really think it was important to have that research assistant support for the child to be able to go through this. And here we really focused on a subset of modules because of the both participant burden. So we didn't, want to put too much on the child, but also focusing on things that we thought we really needed the child self-report. So mood, anxiety, and suicide are all more in the domain of what people might refer to as internalizing symptoms. And we know from the previous literature that parent and child self-report can start to diverge around school age on these things. And so we felt it was really important to get the child's perspective on their symptoms of depression, bipolar disorder, anxiety, and suicide. But we did not assess the externalizing diagnoses in children because of the evidence that kids are not particularly good self-reporters about that. Now, we also wanted information as much as possible about diagnoses of parents and family, because that is certainly important in terms of thinking about familial influences on mental health in children. So we also at baseline administered a modified family interview for genetic studies, or FIGS, that gives us information on diagnoses for first and second degree relatives. We do plan on redoing this again at a later point in time. And we've been under discussion about kind of when is the right time to do that, because you don't want to do it too soon, because you would like for there to be time for new diagnoses to emerge if they're going to emerge, but we don't want to wait too long and sort of miss critical information. All right, so this gives you an overview of the youth KSADS modules. So here is baseline. All of the ones in purple are the ones that are being administered to the youth. So at baseline, like I said, they get mood disorders, social anxiety, generalized anxiety, sleep, and suicidality. At year 1, everything was much shorter. So we just did suicidality and then alcohol and substance use, but only if certain other factors were triggered. And the substance abuse work group can talk about that more so. At year 2, it looks pretty much like year 1, except we added in some subset of questions of conduct disorder and eating disorders that we felt youth would be able to start to report on. And we will continue with that same basic components at year 4, which will be upcoming. And then we will probably add additional modules going out into year 5 and year 6, but those have not been decided on yet. Here's the parent, and same kind of structure, baseline, year 1, year 2, and so on. You can see many more modules are administered to the parents, and more at the longer sessions, which are baseline, year 2, and year 4, and fewer at year 1 and year 3. And again, a pretty comprehensive assessment of externalizing disorders across the different waves based on parent report. We've also added in asking about tic disorders. We do ask about, sorry about that, autism spectrum disorders, but to be super clear, at baseline in year 2, the KSADs did not have a really comprehensive assessment of autism spectrum disorders. So we do not have a good diagnosis of autism spectrum disorders from baseline or year 2. The KSADs, because we were really the sort of beta adopters of it, they now have a version 2.0 that does a much better job of assessing autism and psychosis, and we have switched to that version. And so we will have better, more valid diagnoses of autism spectrum disorders and sort of full-blown psychotic disorders in the out years. But many people have asked me about autism diagnoses at baseline. We do not have a valid diagnosis of autism at baseline. Okay, a few scoring and QC issues that people should be aware of when they access this data and think about using it. What you get when you get the KSADS data is information about symptoms and diagnoses that are coded as present versus absent. We do not have the 0, 1, 2, 3 coding that kind of the diagnostic reports of the KSADs use. In this upcoming release that will come this summer, we will further distinguish between items that were purposely not asked because we go through a screener. If the child doesn't hit on the screener, they don't get the more detailed module. So we're going to distinguish between questions that were purposely not asked versus they were asked and answered no versus missing data. So right now, things that were not asked or were asked and answered no are both coded as zeros. But going forward, we will distinguish between all of those in case people have questions where they want to be able to distinguish between those. I already mentioned this point about the autism-like diagnosis is not valid in KSADS 1.0, but it is improved in KSADS 2.0. We also have noted there was an error in administration where agoraphobia was accidentally not administered for part of year 1. We changed how we launched the KSADs, and that unfortunately wasn't done, but it has been fixed in the out years and will be assessed. This is not a very frequent diagnosis in kids, so we don't think that we've missed a lot of important data here. All right, now categories and diagnoses are important, of course, but we also wanted a very rich assessment of mental health symptoms from a dimensional perspective. So for parents, we have reports on the child behavior checklist, and just a shout out to the ACEBA folks who have been great supporters of ABCD and allowing us to use these measures. That's administered starting at baseline and ongoing over the years. Oops. Sorry about that. We also have a brief measure of mania risk or kind of mania-like symptoms because that's not well captured on the CBCL, called the General Behavioral Index of Mania, starting at baseline. We also added in a very brief version of the Social Responsiveness Scale, which is a measure of autism spectrum symptoms that started in year 1. Then from child self-report, starting at baseline, we do a child modified version of the prodromal psychosis questionnaire brief, and we do that every year. We've already published a number of papers on that. We do the 7-Up measure of mania-like symptoms in kids starting at year 1. And we did the brief, and we have been doing the brief problem monitor, which is a kind of a very brief self-report version of the child behavior checklist. And we started that actually at 6 months. We do it at all the online assessments, and then we do it at year 1 and year 2. We will be shifting to the longer use self-report starting in year 4, which will give us a more extensive self-report of dimensional measures of mental health in the youth starting at year 4. We kept it short before that because of participant burden concerns. And then we also have a brief delinquency measure that we started at year 1 to kind of start to measure from the child's perspective some behaviors that might be categorized as delinquent behaviors. And just a note, we are missing one of those items at year 2. It was a snafu in administration, but that has been fixed going forward. And then lastly, we also have a measure of positive mood or sort of positive affectivity starting at 6 months that we do online and in person from the child's perspective. So we want to kind of to balance all the questions that tended to focus on negative things with things that might focus on more positive things. And then we also have teacher report yearly on the brief problem monitor, which is also a very short measure, again, a subset of the kinds of items that are on the child behavior checklist. And we also have parents report about themselves because we're really interested, again, in the sort of relationship between parent experience and child experience. So the parent who brings the child in reports on themselves, or caretaker, I should say, on the adult self-report, which is like the adult version of the CBCL. And then we also started in year 2 having the person who brings the child in report on the other adult in the household using the adult behavior checklist. So that gives us more information about the dimensional mental health experiences of the child's caretakers. And this just gives you an overview of how this is distributed across the years. So these are the child report measures here. And these are the parent report measures and the teacher measures, baseline, year 1, and so on. So you can see we have the prodromal psychosis scale every year. We have the 7-Up mania every other year. We started with the brief problem monitor here. We continued, but we're switching to the use self-report here. Use self-report contains all these items, so you could have a continuous measure here. Brief delinquency, and so on. And then the teacher measures we're trying to acquire every year. I will say it does get more challenging to do the teacher measures as the kids get older because they start to move into classroom structures where they don't necessarily have a clear homeroom teacher who's very familiar with their behavior, but we have some systematic guidelines for which teacher to select in the out years, as they move into middle school and high school, where they don't necessarily have that one homeroom teacher who, sees them a lot and has a lot of access to their behavior. All right, so we also measure other kind of personality and other related behaviors that we think are really relevant for understanding mental health and also risk for substance use. So the parents report about the child's temperament or personality using the early adolescent temperament questionnaire. And then we also added in a parent report about the child's emotion regulation abilities because that is clearly very relevant to understanding mental health. And then from the child perspective, starting at baseline, we have a very brief measure of impulsivity called the OOPS. This was a modified version, and we've already published a nice validation measure on it. Ashley Watts took the lead on that. And then we've also added in emotion regulation items from the emotion regulation questionnaire in year 3 to get at the child's perspective on their emotion regulation abilities. And again, here's just to give you an overview of this. So the OOPS is getting administered every other year. And then we added in both the child and the parent perspective on emotion regulation starting in year 3. And we've got the temperament / personality measure in year 2. And we're thinking about re-administering it in year 4. There's always this tension, in theory, temperament and personality are stable and you would only need to measure it once. But, for safety's sake, we want to at least get a couple different measures. And especially if you want to look at sort of longitudinal perspectives on behavior, we're trying to get as many measures as we can over at least 3 waves, which is really optimal for being able to look at longitudinal trajectories. And then I also mentioned that we're trying to get measures of stress and life events and also friendship behaviors. So from a parent report perspective, we started asking parents to report on life events that the child experienced starting at year 1. And we have been trying to make sure that we include sort of current relevant types of categories that aren't necessarily in the traditional measures. So for example, we've added questions around a family member being deported, foster care, school shootings, and now COVID-related items. And then we also added in year 3 the parent perception of their own life stress, because we thought that that was probably pretty relevant to the family's experience. And then from the child self-report perspective, we also asked the child to report on their own life events starting at year 1, again, with these added kind of relevant measures around deportation, foster care, school shootings. We also, starting in year 2, asked them about their experiences of peer victimization as well as perpetration. So around things like relational aggression, other kinds of bullying experiences, either that they may experience or they may perpetrate. We've also been asking about cyberbullying experiences, again, both victimization and perpetration. And we ask them about their friend structure. So we ask about the number of friends who are boys and girls, the number of close friends who are boys and girls. And clearly this is important because all of these sort of peer relationships and social relationships are really important for understanding mental health over time. And again, here's the overview of when these different things are measured. Started life events in year 1, and we'll be continuing with that. Peer experiences in cyberbullying in year 2, and we'll be continuing with that. And an assessment of parent life stress in year 3, and we're hoping to be able to repeat that in year 5. And I should note, I only put year 4 on here because we have not planned year 5 yet. So telling you about year 5 would be speculation. So I'm showing you the things that we've definitely decided on. So a few things to think about when accessing and using the mental health data from the ABCD study. You will find quickly that parent and children frequently do not agree. This is a pretty common finding in the literature, so this is not unique to the ABCD study. And we have been talking about ways in which you might think about developing integrated diagnoses, because you have both parent and child report. I think a lot of people have been sort of doing the sort of quick and dirty approach to that, which is saying, I'm interested in diagnoses of depression in children, and I'll say that the child is depressed, if either the parent or the child reports things that meet criteria. So that's certainly one way of doing it, but one could think about more complex or sophisticated ways of integrating those two reports. But I think it's also important to think about the fact that different reporters may be better for reporting on different kinds of behaviors. So, we do ask parents much more extensive information about externalizing symptoms. The kids do report on some of that from like the brief problem monitor, but you may want to focus on teachers or parents for those kinds of behaviors. Whereas kids may have access to internal life experiences, kind of internal experiences more so for internalizing. So you may think about wanting to focus on child self-report for those. It'll be important to look at consistency over time. We have been starting to look at this, and there certainly are, there is an element of test-retest reliability and consistency, but reports do change over time. And some of that is because people's actual experiences are changing over time, but some of it is also going to be people don't remember what they were experiencing at a previous point in time. So be prepared for that. Like I mentioned already, it's kind of an open question when we should think about reassessing things like family history. We're probably thinking 4 or 5 years in is good, because as the kids get older and older, they're going to be able to do more and more things on their own without their parents necessarily involved. And so we kind of want to wait as long as we can without losing good parent reporters for some of those things. I have just a brief amount of time, and I thought I would just share a few early results just to kind of see what sort of data is there. And I'm going to focus on 2 domains that were of particular interest to the National Institute of Mental Health, and that is suicide and psychosis. And I focus on these because I think there was some initial skepticism about including these things, some concerns that suicide-related behaviors wouldn't at all be present in 9 and 10-year-old children, or the thought that maybe you couldn't validly assess psychotic-like symptoms in kids. And I think the early data suggests that neither of those concerns was really warranted. So first, let me show you data from a paper that's in press now on the rates of suicidal ideation and behavior in 9 and 10-year-old kids. So if you look at passive ideation, which is just things like wishing I was dead, if you look in the baseline sample, which is 11,800 kids, we see 763 kids who report on that. Now you might say, what's the population estimate mean? Well, we can use what we call propensity weighting to kind of match our sample to a nationally representative sample and say, if you extrapolate it out, what would that suggest about the population estimate of kids age 9 and 10 in the US who might be experiencing passive ideation? And this would suggest that about 6% of kids, or almost 500,000, might be experiencing this. If you look at active suicidal ideation, that's non-specific. So I would like to kill myself, but I don't have a plan. We see fewer kids, which is good. But again, if you look at a population estimate, close to 300,000, about 4% of kids. If you look at more specific ideation about suicide, where they might have a particular idea about how they would do it, again, fewer kids, fortunately, but up to 2% of the population. And if you look at kids who say that they've already made an attempt, or parents that said their child had done something that they think was a suicide attempt, we still have over 100 kids who are saying that, a non-trivial number. So this does suggest that definitely there are 9 and 10-year-old kids who are already having these kinds of thoughts. And we know from other data that early suicidal ideation does predict later suicidal ideation. So it is definitely important to be looking at this early. If we look at factors associated with suicidal ideation and behavior, interestingly, gender is, but not in the way that you would think. We actually see higher rates in boys than girls for most things other than attempts. That will switch later on. We do not see strong associations with race, but we do see strong associations with a family history of depression. So it is much more common across all the different categories amongst children who have a family history of depression. And this is also true if you look specifically at a family history of suicide. If anything, it's even stronger. We also see that financial adversity is associated with higher rates. These are odds ratios. You see a significant difference between those who endorse that thing versus those who don't. And so you have greater financial adversity among those who do. You see greater internalizing problems among those who do. You see greater externalizing problems. You actually see less parental monitoring associated with greater rates of passive SI, nonspecific suicidal ideation. So if you look here, right, parental monitoring goes down as you look at, like, say, kids who have suicide attempts. And kids who have these different experiences also report less caregiver acceptance and greater family conflict. So kids who are having passive and active SI and attempts and non-suicidal self-injury have greater financial adversity, greater internalizing and externalizing symptoms. They report lower parental monitoring, lower caregiver acceptance, and greater family conflict. Turning to assessing psychosis risk in children, we used 21 questions from the psychosis questionnaire brief, which had been adapted for children from adults. It took questions that, in adults wouldn't be clear to kids and tried to use more kid-friendly language like unusual powers, things magically happening. Did you feel confused because things didn't seem real or imaginary? These are just a few examples here. And then if a kid said something happened, we asked them whether it bothered them. And if it bothered them, we asked them to tell us how much it bothered them using this scale. So we can get a distress measure as well as just a frequency measure. Let me show you some of this data. So if you look at the average score as a function of race, you see, unfortunately, that kids whose families report them as being African American, Hispanic, or other races show significantly higher distress scores and total scores compared to kids whose parents categorize them as Caucasian or white, and that is consistent with the adult literature where you also see higher rates. And I should note that we did measurement and variant analyses, and the scale is invariant across race and gender, suggesting that it is measuring the same thing across races and genders. We also see that kids with a family history of psychotic disorders show higher PQB distress scores than those who don't. And at least, and you don't see that less with family history of depression and mania. We also see that kids who have speech delays and motor milestone delays show higher PQB distress scores, and that is consistent with the adult literature suggesting that you are more frequently have speech and motor delays among people who develop psychotic disorders in adulthood. We also see that you have reduced IQ, both fluid and verbal IQ, amongst children who self-report highly severe psychotic-like experiences, again, consistent with the adult literature on psychosis. And if we look at more specific cognitive functions like working memory, processing speed, episodic memory, we see that all of these, including reading, are also associated with higher PQB distress scores, again, consistent with the adult literature. And then I'll just tantalize you with a little brain data. So in adults with psychosis, we see disruptions in functional connectivity among brain networks that are thought to be important for cognitive function and cognitive control or executive control in particular. And if we look at that, we look at various brain networks. You can look at particular brain networks like the frontal parietal network in yellow here, which includes the dorsal frontal and dorsal parietal regions. You can look at the cinguloopercular network in purple, which includes the dorsal anterior cingulate and the anterior insula. And both of these are networks that are thought to be really important for executive function and cognitive control. And if you look in adults with psychosis, we see reduced connectivity among these networks. Well, if we look, and you could have a comparison network that isn't thought to be impaired, like the auditory network in this kind of light purple. And if we look in kids in the ABCD sample, we actually see that greater PQB distress scores are associated with reduced connectivity among the cinguloopercular network. Not true for the frontal parietal network, not true for the auditory network. So hints again that this measure of psychotic-like experiences in kids is capturing something that is related to the kind of psychosis spectrum. Although fortunately, many of these kids will experience this transiently and it will go away. And so now we are starting to look at what happens for kids who have these kinds of experience sustained over time. All right, so let me summarize up. Suggestions for addition in out years are always welcome. We can't add everything, but we're always interested in hearing suggestions for additional things. Longitudinal analyses are really going to be the next key step. This next wave of data release are going to have data over multiple waves, so you can start to look at longitudinal outcomes. And we will be shifting more and more of the assessment of mental health to the youth as it becomes developmentally appropriate to do so. And we think that these trajectories are going to be increasingly important as we have more repeated measurements of mental health. And I should note that we have also launched assessments of COVID-19 related experiences, including mental health experiences, which we think will be highly informative, both as predictors of responses to the pandemic and also whether our baseline measures help us understand the consequences and the responses that youth have. So I'll just end here saying, please send your questions here if you have them, and thank you for your attention today.
I'm going to be speaking today about the neuroimaging processing pipelines used for the Adolescent Brain Cognitive Development Study, or ABCD. I'll be talking first about the processing pipeline components, then I'll discuss the quality control procedures, data sharing, between scanner differences, and information resources that are available. So the processing pipeline uses a software package that we've developed in-house that we call the multimodal processing stream. The processing pipeline is described as used in ABCD release 2.0/2.0.1 in this paper that's referenced here. So we conceptualize different stages of processing. The first stage involves the unpacking and conversion of DICOM files, the raw images. These converted files are then processed through distortion correction and motion correction, as well as between modality registration. The next stage is brain segmentation, where the whole brain is segmented, the cortical surface is reconstructed, and white matter tracts are segmented. The analysis stage is for individual subject level analyses, and that includes structural MRI morphometry, diffusion MRI modeling, resting state fMRI correlation analysis, task fMRI event-related analysis, and volume and surface ROI averaging. Finally, the summary stage collects all of the measures for all the regions of interest and then compiles across subjects as well to produce data in a tabulated form or a spreadsheet. Structural MRI includes 3D T1 weighted and T2 weighted images. The structural MRI processing includes gradient nonlinearity distortion correction, registration of the T2 weighted images to T1 images, intensity non-uniformity correction, resampling, and brain segmentation and cortical surface reconstruction using FreeSurfer version 5.3. The structural MRI derived measures include morphometric measures like cortical thickness, area, volume, and sulcal depth. And we also produce image intensity measures derived from the T1 and T2 weighted images for cortical gray matter and the pericortical white matter, as well as the contrast between them. We calculate average measures for cortical surface ROIs using standard FreeSurfer parcellations and subcortical ROIs for intensity-based measures and volumes. Diffusion MRI uses two different modeling methods, diffusion tensor imaging and restriction spectrum imaging. First, the diffusion MRI pre-processing includes head motion correction, correction of spatial distortions, robust diffusion tensor estimation, registration of the T2 weighted B equals 0 images to the T1 weighted image, and resampling into a standard orientation with 1.7 millimeter isotropic voxels. So diffusion MRI measures include two different modeling methods, diffusion tensor imaging and restriction spectrum imaging. For the DTI derived measures, we have two different models. One is using the inner shell, or just the B values less than or equal to 1000. And we also have a model using the full shell or all of the B values. Restriction spectrum imaging takes advantage of the multiple B values in the diffusion MRI sequences used for ABCD to separate restricted and hindered diffusion within individual voxels, corresponding to intracellular and extracellular signal fractions. We average the DMRI derived measures in regions of interest, including white matter tracts, subcortical gray matter structures, and cortical parcellations, both for cortical gray matter and pericortical white matter. Resting state fMRI begins with pre-processing steps that are common to both resting state and task fMRI. So these steps include B0 distortion correction, gradient nonlinearity distortion correction, head motion correction, and registration to the T1. There are some resting state specific pre-processing steps, and that includes the removal of initial volumes with the number of TRs dependent on the scanner platform, filtering the data, meaning normalizing and detrending time courses, regressing out signals correlated with motion and global signals, the censoring of frames with excessive motion, and bandpass filtering to isolate the slow wave oscillations. Then finally, the time series data from voxels are sampled onto the cortical surface. We apply a seed-based correlation analysis where average time courses are calculated within functionally defined parcels and subcortical ROIs. The parcellation uses this so-called Gordon parcellation that is derived from resting state correlations and includes the definition of several networks among the different parcels. So we calculate pairwise correlations between all of the ROIs. and then calculate correlation within and between the predefined networks. Finally, we calculate correlations between each subcortical ROI and each network. Task fMRI for ABCD includes 3 different tasks. First, the monetary incentive delay task, or MID, stop signal task, or SST, and the emotional end-back. These 3 tasks are designed to elicit activity related to executive control. Task fMRI analysis includes some specific pre-processing steps, including the removal of initial volumes, the normalization and demeaning of time courses, sampling the time series data onto the cortical surface, and then calculating ROI average time series for subcortical segmentation and anatomical parcellation ROIs. The general linear model is used, implemented with AFNI's 3D deconvolve. Nuisance regressors are included to model the baseline cubic polynomial and motion. Time points with excessive motion are censored. And the hemodynamic response function is modeled as a gamma function with temporal derivatives. For the MID and SST models, events are modeled as instantaneous, whereas for the end-back, the duration of cues and trial blocks are modeled as square waves convolved with the gamma function. GLM beta coefficients and standard errors are calculated for regions of interest, as well as vertex-wise and voxel-wise calculations. Censoring for outliers is applied for the task contrasts with the root mean squared of the SEM values across the whole brain greater than 5% signal change. Our quality control procedures have evolved a bit over time. In the last release, release 2.0/2.0.1, all of the images were reviewed at the for data quality as they come in, as they're collected and received at the Data Analysis Informatics Resource Center. Images are reviewed for various artifacts, and images with severe issues are excluded from further processing. We also perform post-processing image and segmentation review. For the 2.0 release, we included FreeSurfer QC for all baseline visits and DMRI QC for half of the baseline visits. We also published imaging inclusion criteria that were included in the release notes and in the processing pipeline paper I mentioned earlier. Some new things to be included in the release 3.0 or changes for manual post-processing QC. Now we are including QC for FreeSurfer for all of the baseline and the 2-year that are included in the 3.0 release. And we'll be including about 20% of the diffusion and fMRI data sets with the post-processing QC. We're also including new automated post-processing QC metrics that I'll describe a bit in the next few slides. And we'll be including some simplified imaging inclusion flags that facilitate the selection of data for analysis. So the manual post-processing QC involves 2 different types of quality control scores. The first is an overall QC score with a value of 0 meaning fail or reject, and a value of 1 meaning pass. We also make qualitative scores according to different modality-specific common problems that we look for. We give them a score ranging from 0 to 3, with 0 meaning no problem and 3 meaning severe enough to fail. For FreeSurfer QC, some of those common problems that we look for are motion artifact, white matter underestimation, peel overestimation, intensity inhomogeneity, and susceptibility artifact. For diffusion MRI, those common problems include residual B0 distortion, registration to T1, image quality, brain cutoff, and fiber segmentation. For fMRI, we evaluate residual B0 distortion, registration to T1, image quality, and cutoff. So automated post-processing QC for the different modalities is something that we've been working on in this new release. So for FreeSurfer, we will be including the number of topological defects for a given subject's cortical surface reconstruction. This reflects on the data quality and also can reflect a bad cortical surface reconstruction. For diffusion MRI, some of the key measures include a field of view brain cutoff measure, shown here on the top right, and a registration to T1 evaluation shown on the bottom right. For fMRI, we also evaluate the FOV brain cutoff and the registration to T1. And for T2, we calculate metrics related to the registration to the T1. Here's showing some examples of the field of view brain cutoff measure for a case with dorsal cutoff and a case down here with ventral cutoff. So the measure, the cutoff measure, is calculated as the percent intersection of a brain mask with the frame borders. To evaluate the registration to T1, we focus on a small window around the corpus callosum. We reverse the contrast of the T2 weighted images so that they best match the T1 image, and then register those two images in that small window to estimate both translation and warping to capture any residual distortion in the fMRI or diffusion images and to capture problems with the registration. So here I'm showing a bit of a work in progress where we're trying to detect anomalous data and distinguish it from normal scans. We're using a statistical machine learning based approach. So we start with performing manual QC in about 10% of the diffusion and fMRI data sets. We then use those manual QC scores to train a Bayesian classifier. We then predict the manual QC scores based on one or more automated QC metrics. Additional manual review is then done for data classified as problematic. So here's showing some examples of images detected using this approach. On the left for images with substantial brain cutoff, and on the right, images with problems with the registration of the T1. So as I said, in the next release, we're also going to be including a set of imaging inclusion flags. These are based on modality-specific inclusion criteria. These are sets of variables with threshold criteria. For example, the number of time points after censoring in resting state fMRI data, or behavioral performance in the task fMRI, or post-processing QC for free surface reconstruction. These inclusion criteria have been implemented in DEEP and NDA to select data for analysis. And DEEP is the data exploration analysis portal that will be described in another talk in this symposium. And the NDA is the NIMH data archive, and I'll talk more about that in the coming slides. These inclusion criteria can be used by investigators as templates for their own customized inclusion criteria or used as recommended. The imaging inclusion flags that we're creating for the next release are pre-computed combinations of these inclusion criteria, where we'll have a binary flag, a 0 or 1 for each modality, whether we recommend including that data in analyses. So an example of how that works in DEEP is with a drop-down menu, a user can select the set of recommended DMRI data sets and then perform their analyses on that. And over on the right, showing a schematic of the different inclusion criteria and how they're applied and conceptually as different filters, removing data that should be excluded for various reasons. So in this one is showing some of the fMRI inclusion criteria that were published in the release 2.0. And these include a few extra variables, such as requiring a valid E' file, the behavioral and stimulus file from the task fMRI, or requiring a certain amount of data after censoring for motion. So anyone who is using the imaging data from ABCD should be aware or become aware of the between scanner differences related to the scanner manufacturer. So for example, T1 gray-white contrast is lower in Siemens scans, whereas the thickness estimates are lower for GE and area is slightly higher for GE. Similarly, there are differences between scanner platforms for the diffusion measures, and I'm not showing it here, but also for the fMRI. I do want to focus on, for the T2 weighted images, pointing out that not only can there be differences between the scanner platforms, but also between specific scanner instances, say within the Siemens scans. So for the T2 weighted images, this is caused by global differences in scanner parameters at those sites. So for this reason, we recommend including as a covariate the scanner instance in any analysis using the imaging data. So public data sharing is done through the National Institute of Mental Health Data Archive, or NDA. So if you go to the NDA website, you will find a button that says ABCD, and if you click on that, it will take you to this page where you'll find more information about ABCD and different links that you can click on. So if you click on this fast track release info, it'll take you to this page. So fast track data is a mechanism for sharing the raw imaging data in DICOM form relatively quickly after data collection. So approximately within a month of the data collection, it will be shared on NDA for users to access. Note that this data is not completely curated and is released very quickly. And so this is to distinguish it from the data that's included in our annual curated data releases. So that includes minimally processed data for all of the structural, diffusion, and fMRI images with the various processing steps applied, as well as additional files necessary for individual subject level analyses for that modality. Also included in the annual curated data releases are tabulated results. For the imaging, that includes structural MRI, diffusion MRI, resting state fMRI, task fMRI, quality control measures, and metadata. So these releases, these annual releases have started in 2018 with release 1.0, and release 2.0 came out in the summer of 2019. So this first release had about 4,500 of the baseline subjects, and it was done while collection was still ongoing. And release 2.0 was released with the full baseline sample of about 11,800 subjects. For the upcoming release 3.0, which will come out in August 2020, there will be the full baseline and half of the 2-year follow-up. And we will include cross-sectional metrics only for release 3.0. For release 4.0, we'll be including the complete baseline and full 2-year follow-up data sets. And we'll be including both cross-sectional and some longitudinal metrics. And that will be expected to be coming out in August of 2021. Some of the new things that are coming in release 3.0 include, as I said, an increased number of data sets. So baseline data for about 11,800 participants and 2 time points for about 6,000 participants. There are some fixes and improvements made to the processing steps for fMRI. We fixed an incorrect B0 distortion correction that was being applied to the Philips data. And we've improved the diffusion and fMRI B0 correction by using FSL's top-up. As I've mentioned, we are now going to be including imaging inclusion flags with updated inclusion criteria. These will incorporate manual post-processing QC, automated post-processing QC, and additional variables. Some of the areas for future development that we'll be working on in the coming years include, for data sharing, we'll be including raw imaging data in the BIDS format with Nifty files in our fast track data sharing. We'll also be sharing derived data in BIDS format, for example, FreeSurfer outputs and task fMRI contrasts. We'll be working on our pre-processing steps, including enhancements of the diffusion MRI processing and to the fMRI motion correction. Brain segmentation and analysis will be improved in certain ways. For example, we'll be upgrading to FreeSurfer 6.0. We'll also be updating the RSI analysis for diffusion MRI and including within subject longitudinal change metrics, particularly for structural MRI and eventually for diffusion MRI as well. And as I mentioned before and showed with some figures, there are known between scanner differences in the existing data sets. We'll be working on implementing between scanner harmonization to correct for some of those differences at the level of the individual images. For structural MRI, this includes a histogram-based contrast correction for the T1 and T2 weighted images. And for diffusion MRI, this will include a model-based correction of scanner differences, with the hope to reduce any scanner biases in the derived measures. So there are a number of information resources available. First, to get help accessing the data from ENDA, you can contact the NDA help desk and request access or get help with packaging and downloading the data from NDA. And as I mentioned before, you can refer to this paper that was recently published that describes the processing pipeline in detail. In addition, there are release notes associated with each release and additional files that are available through NDA on the ABCD collection page. You can also receive announcements from ABCD by subscribing to this email list. Once subscribed, you will receive announcements that include notes and alerts about upcoming releases and known issues. There are a number of ABCD associated tools that you can access through GitHub on the ABCD study page. For example, there are tools to extract timing information from ePrime files for the task fMRI. You can find deep code there, as well as packages for converting the fast-track DICOMs to NIfTY in BIDS format, and a customized HCP pipeline for ABCD data. So you can find more about those packages by going to the GitHub page. We also have a multimodal processing stream Docker that you can access and use on ABCD data. That's accessible from Nitric. And so currently the version that's available is consistent with release 2.0/2.0.1. And after release 3.0 comes out, we're planning on updating that Docker with the latest version. If you have any questions about the processing pipelines or anything I've talked about, please go to this web page and enter in your questions so they can be addressed in the live session. I'd like to thank a couple people who were instrumental in preparing these figures. First, Payam Saizan, who has been working on the automated post-processing QC metrics and the machine learning classifier. And also Sean Hatton, who's helped with the between scanner difference analyses and pulled together the information resources. Finally, I'd like to thank the ABCD families and the child participants and the number of different funding sources. Finally, if you'd like to learn more about the ABCD study, please go to abcdstudy.org. Thank you.